Good morning. It's a pleasure to be here virtually. I'd like to thank the organizers for inviting me. The work that I'm going to present is principally the work of Dr. Han, recent graduate of our lab, pictured in the center. We have a small lab, a lot of collaborators, and a very small amount of support. This is my disclosure slide. The work I'm going to present has recently been published in AJNR. And we're going to talk about primary brain cancer, glioblastoma. The goal is to develop and validate MR biomarkers that can allow us to predict treatment response and prognosis. Principally, we intend to use these in therapeutic trials of novel therapies, including immunotherapy. The data type is post-contrast brain T1-weighted MR images. The analysis is radiomics, augmented by deep learning. And the gold standard is patient overall survival. The paper that I'm presenting used convolutional neural networks to extract a large number of so-called deep features that we combine with the radiomics features, which are derived in a heuristic way from the same image data set. The combination of these so-called handcrafted radiomics features and the deep features constitutes a rich data set, and we try and use this to model, produce a predictive model of patient overall survival. There are three data subsets in our paper. The first is 50 patients from our institution. All of them had 2D spin echo T1 weighted post-contrast images because this is a relatively older data set. A second data set is downloaded from the internet. It's 128 patients, also with glioblastoma. Some of these patients had 2D spin echo images, exactly the same as our institution, and some of them had 3D FPGR higher resolution images. These come from multiple institutions. Subsequently, we created a combined data set where we handpicked the 58 TCGA patients that had the identical spin echo 2D T1 weighted images as our patients, and combined them with our patients to produce a data set from multiple institutions that was technically homogeneous. And I'll explain why in a minute. The demographics of these patients and their clinical characteristics are fairly similar. The patient treatment protocols are also quite similar. Both data sets are very heterogeneous in the sense that patients underwent standard Timidar chemoradiation in most cases and a large variety of other treatments. I don't think there's a systematic bias between these two very heterogeneous real-world clinical data sets. Preprocessing is fairly standard for use of convolutional neural networks. We do intensity normalization and noise removal, gray level quantization, and 2D in-plane resampling to get uniform voxel size. I won't go into the architecture of the neural network. I'm not an expert, and I think you probably heard a lot about these. Basically, we have a neural network that has been developed using very, very large numbers of images on the internet to classify photographic images. We're not modifying this in any way. We're just inputting our MR tumor patch, which is a subsample of our image that contains the area of abnormality, into the input layer, running it forward through the neural network, and then extracting the nodal weights from the last two fully connected layers and using them as so-called deep features. So we're not actually interested in this case in the output of the classification produced by the neural network, but we're using its fully connected layers as a source of additional image features. We combine these with the handcrafted features and then perform more analysis. Basically, the architecture here is fairly standard. I won't dwell on this. The experimental design is a little tricky to explain clearly. We take the very large number of features on the substantial number of patients in the full data set and do feature reduction. This is essential because the number of features here, more than 8,000 deep features and 300 handcrafted features, is larger than the number of patients in the data set. After that, the reduced feature set is run through a leave-one-out cross-validation scheme in which sequentially each patient is, in one iteration of the loop, left out as a test patient, and the remaining patients constitute the training set, which is input into the so-called inner loop. The inner loop consists of additional feature reduction and then Cox modeling. And a 10-fold cross-validation scheme is used to optimize the penalty terms in the Cox model. The output of the inner loop is an optimized predictive model. That model is applied to the held out patient to generate a prediction of that patient's overall survival as longer or shorter compared to the median overall survival of the patients in the training set. As all the patients are iterated through the leave-one-out cross-validation outer loop, these predictions are concatenated into a prediction array, which we can then characterize, use to characterize the pipeline's accuracy for prediction. The feature reduction methods include median absolute deviation in the outer loop, where we remove features with low variation from the full data set, and then in the inner loop, concordance index, where we characterize the concordance of each feature with overall survival and remove those features which have a concordance below a set threshold. Subsequently, we do a Pearson correlation coefficient between each pair of features to identify features that are highly correlated with each other and remove the feature that has the lower C-index with overall survival. The output, I think, is quite interesting. From our data set, the most homogeneous, one institution, one technical type of T1 weighted image, we get a very good separation of the two groups, showing that the pipeline can accurately predict shorter and long-term survival. We get a hazard ratio above 3 and a very low p-value. When we retrain the entire pipeline on this more heterogeneous multi-institution data set from TCGA, we still get good performance, which is encouraging. We are able to separate the two groups with a statistically significant p-value, but the hazard ratio is not as high and the p-value is not as low. We then constructed and retrained the pipeline on our multi-institution but technically homogeneous data set to see if we could rescue the performance. The hazard ratio did not improve significantly, and the p-value did not improve. And this is interesting because it suggests that the identifiable contributor to data heterogeneity, meaning the use of high-resolution 3D images or 2D thick section images, does not appear to account for the difference in performance. And something else about the heterogeneity of this data set related to some unknown factors that are likely based on the fact that these are required at different institutions creates a degradation of pipeline performance despite retraining the pipeline when we apply this pipeline to more heterogeneous data sets. I'm also going to point out that another weakness of the radiomics approach is that both the handcrafted radiomics features and the deep features are not interpretable to human beings. Even the handcrafted features, aside from a few that are non-texture features, volume, size, and a few that are based on the histogram and are somewhat understandable, are completely incomprehensible to human beings. As a result, none of the radiomics features can really be understood in terms of what they mean. So our conclusions are that the VGD19 weights generated from the post-contrast images complement the radiomics handcrafted features in prediction of overall survival. And using this combination, we can separate short and long-term survivors. That's good news. However, the meaning of the deep and the high-order radiomics features is unknowable. So this contributes little or nothing to our scientific understanding of the pathophysiology or how the images, what the information in the images is that predicts survival. This is what we call the interpretability problem. Also, despite translating the pipeline, meaning completely retraining this, rather than trying to translate the model, and carefully matching the data, our performance still declined with increasing heterogeneity of the data set. This is what we call the generalizability problem. I think it's closely related to the interpretability problem because we don't know what the neural network is paying attention to. That being said, the next step toward clinical use is prospective testing on a large, independent, technically and clinically distinct data set to again see how this affects our performance. There are various optimization targets. We can pick different neural networks, especially those trained on MRI, choose different features from different layers, add additional pulse sequences, etc. Nevertheless, overall, I think the most interesting implication of this work is that the approach works, but that we cannot, with confidence, predict how it will work on data sets other than that on which it was developed. Thank you.